Answering MCAT questions in under 60 seconds. In a study of bacterial gene regulation, researchers examined a transcriptional activator protein, ACTR, which binds upstream of the LACX and LACY genes. When lactose is present in the environment, ACTR expression increases, and ACTR binds DNA to promote transcription of both LACX and LACY. A mutant bacterial strain lacks the ACTR gene. Which of the following best predicts the outcome in the mutant strain upon lactose exposure? So for one thing, ACTR appears to be some form of an enhancer, right? Which is basically going to promote expression of the genes of the lac operon right here, or whatever this operon is, but it sounds kind of like the lac operon, right? And remember that with the lac operon, lactose, when it is present in the environment, right, it is going to bind to a repressor molecule that is just at baseline bound to the lac operon and blocking transcription. And so lactose binding to that repressor will cause it to detach and allow for transcription to proceed. However, to amplify that transcription, you usually need some sort of enhancer molecule, in which case ACTR seems to be one that is acting as an enhancer. So A, transcription of LACX and LACY remains unchanged compared to wild type due to redundancy in the regulatory pathway. So that's wrong, right? Because if ACTR is something that's enhancing expression, then even though lactose is present and kind of disinhibiting expression of these metabolic genes, right, you're only going to get some basal level of expression, but in order to really get high levels of expression, you're presumably going to need this enhancer. So you should expect lower levels of transcription in this mutant. B, transcription of LACX and LACY increases despite absence of ACTR, since lactose directly promotes RNA polymerase binding. So while it does promote RNA polymerase binding, without the enhancer, you're not going to have an increase in transcription, okay, relative to when you have both present, right? So that doesn't make sense. And then C, transcription of LACX and LACY is significantly reduced because ACTR is required to activate their expression in response to lactose. And that sounds correct, okay, right? Because again, while lactose being present is going to promote transcription, you're going to enhance that and really elevate it when you have the enhancer molecule present. So that sounds right. And then D, transcription of LACX and LACY increases slightly due to increased lactose metabolism in the absence of ACTR repression. And again, that's wrong. Like you wouldn't have increased lactose metabolism in its absence. In fact, you would have lower levels, right? So C is the right answer.